Well, thank you very much for the invitation today. I took the liberty of modifying the title that was given to me. We didn't actually work to obtain the requirements with the FDA. The requirements were given to the banks, but we did work with the FDA to meet them. So I'll walk you through that, if I find how this works. So as a background information, the FDA has started commenting on cord blood a while ago. The first publications of the Federal Register was in 1996, and then in 2007, clearly positioning cord blood as a biological product in those publications. I want to note that our bank, which is the National Cord Blood Program, has been in close collaboration with the FDA from very early in the beginning of banking, and we actually did get a FDA IND back in 1996 for cord blood, and I will come back to that again later. But it wasn't until 2009 when the Federal Register and the FDA made their position very clear that cord blood is a biological product, and that banks have to work and distribute cord blood either under an IND, an investigation new drug application, or under Biologics license. So essentially, CBER encouraged sponsors to send their applications in, and with this publication and actually the guidance for industry that came for the banks, the clock started ticking, so to speak, from October 2009 until October of 2011. So there was a 2-year interval in which it was recommended by the FDA that banks have to apply for a Biologics license. And we started. And it's important to sort of keep some terminology into mind because this was new. The regulatory terminology was a little different than what the banking community was using. So given the 2 guidance documents, the one for the license, which came out in October of 2009, and then at the same time, there was a draft document for the IND, which got finalized in June of 2011, so within 2 years. Cord blood is not just called cord blood, but hematopoietic progenitor cells cord, and this is the minimally manipulated product. And the term manufacturing got into cord blood banking, and with manufacturing, we mean all steps from the collection until the release of a product to search. So everything between collection, processing, testing, freezing, cryopreservation, storage, until the product is released to search. And that has to be covered under the term manufacturing. And then the cord blood products are divided now in 2 groups. One is the licensed product, a product that has been licensed by the FDA, which is considered as a prescription drug. Licensed products from the FDA have given indications, so a product is licensed for a specific list of indications. But because it is it's a prescription drug, it can also be used off-label. On the other hand, the non-licensed products, now called IND products, they're investigational, and the new regulations require an informed consent from the patient that has been approved by the hospital's IRB prior to using an investigational product. I will remind you that we are using cord blood in unrelated transplantation since 1992, and these products were investigational all along, but at the time we didn't have to use a product-specific consent. However, with the new regulations of the FDA, all IND products have to have, in addition to the treatment consent, a separate consent for the product. So the Biologics license application was a pretty big adventure that we undertook, and it is a very long document. This is the table of contents that goes and goes and goes, and the point of this slide is to show you that the actual sheer amount of work that we had to do was enormous. Besides the load of the work and the amount of the work, there was an educational process that needed to take place. We were not accustomed to the regulatory terminology used in the guidance document. If anything, the guidance document is a very dense document, and each word or each phrase in there leads to another set of regulations and other documents that one has to review. So it ended up being quite an educational process for the banks to go through this and to decide how to apply their own practices and techniques to meet the regulations. The timeline for our bank is shown here. It's important to know that a lot of the work was done before 2009. It's on this side of the bar. So there were a lot of meetings with the FDA as preliminary meetings where we discussed processes and there was back and forth information. Then we moved to the new GMP facility in 2009 and then spent the whole year in 2010 by putting together the work and writing actually the very big document of the BLA that ended up being, as you see, 2 gigabytes of an electronic submission. This was submitted in January of 2011. The FDA has a process which reviews the original document and then files it, which is in the regulatory terminology means they accept this document, this is adequate, and then the process starts. So the first step was to schedule the facility inspection, which happened in April, and then all the red spots that you're seeing on this timeline is further amendments that we had to submit to the FDA, secondary to letters or telephone conferences and requests. So there was a lot more of exchange of information. There were things that needed to be rewritten, redone. A lot of studies had to be repeated or modified, but the process was going on over these months. And then finally, in November of 2011, so at the end of this timeline, there was the license. Just a few things. I know you may not be very familiar with a biologics license. These are some of the areas that the banks have to work on. It's quite a long list, and I don't have the time to explain a lot of this, but I'll show you an example. But there's things that have to do mainly with the manufacturing of the product, with the labeling and the testing. All the assays that are used in the bank and the testing facilities have to be validated and reviewed by the FDA, and so do the information technology systems. The facility itself is a major point of the inspection, and then there's a major emphasis on the quality unit of the bank and the quality release of the product. For the banks, one of the areas that was a lot of work was creating a batch record. You have to consider that some of these regulations come from the pharmaceutical companies. However, cord blood banks are not pharmaceutical companies. They don't have the same manpower or resources, and the batch is something different. In a cord blood bank, a batch is a unit. So we sort of had to come around all these regulations and come up with ways that we could meet them, but also not change our practices. One of the areas that several banks, I'll give you an example, several banks have struggled is what is called the stability studies. So this is clinically relevant, and this implies the storage time of the transplanted units. And as you see in this slide, from the units that we have provided for transplantation from the National Cord Blood Program, several of them have been in storage for a very long period of time, and they worked well clinically. Well, we have to prove that with validation assays, and this is becoming very important for the FDA because we have to assign an expiration date to the licensed product. So the way we went around this is we designed stability, and we actually improved the stability evaluation we were doing before. And we ended up proposing that we would do annual stability studies and then extend the expiration time by a year for all the units in the inventory. I don't have time to explain to you, but there are 4 groups because we have 4 processing methods in the inventory. The last one is the one that we applied for license. However, when we do stability and evaluation of the inventory, we take the entire inventory into consideration. Therefore, we evaluate the same way now the license units and the IND units. And then each year, the expiration date will extend, which is important because it has to be on the license unit label. So this label is generated at the time of release of a product. It cannot be generated earlier because we don't know what the expiration date it is, since there is a process that we evaluate annually. Again, this is different than the pharmaceutical companies. We had to come up with stepwise labeling and partial labels so that the name of the product goes on and the partial label gets released at the time that the product is shipped. And I'm sure other banks have had similar issues and have come with similar ideas. As I mentioned before, a major part of the inspection is the GMP facility, so I will take you through it briefly. This is the GMP facility at the National Cord Blood Program. The cord blood units come from the receiving area and first go into a room which we call accessioning, where all the information of the unit is entered into the computer. And then they entered the GMP, which is the processing area. This is the processing area. We are using a closed automated system, therefore processing is in the open air, not in hoods. And after processing, the different components pass through windows into the cryopreservation room and then into the freezer room for cryostorage. And these are the bioarchive freezers. So this is the way the units move. Samples come out in the pre-processing and post-processing samples pass through windows into the testing laboratories, and then there's a separate corridor for personnel that enter the facility. I showed you the list of things that we had to work on for the license application, and this all had to do with the manufacturing of the product and the testing. To our surprise, and at some point we were asked for information on product efficacy. That was not clear from the beginning. The guidance document did not discuss efficacy, and the idea was that given the data that had been provided to the FDA all these years and were part of the public file, the FDA docket, that efficacy had been discussed and decided by the FDA, and therefore they would go for the license. However, there's something to consider. The FDA docket data, a significant proportion of which was provided by the National Cord Blood Program, dealt with units and transplants during the first 2 periods of manufacturing and transplants performed with the years between 93 and 99. The new product that we were applying for license was produced with the 4th manufacturing period, 2006, and clearly a lot has changed in product manufacturing, but also in clinical studies and clinical transplantation. So we didn't feel that the docket data were very representative of the field right now. To discuss this, we were asked to present to the FDA Advisory Committee. The FDA goes through an extended advisory committee for every new product of its kind to be licensed. And since we are the sponsor of the cord blood, and this was the first product to be licensed of its kind, we were asked to present safety, efficacy, and we touched upon the indications. And the advisory committee meeting was public. It was a public meeting. It's open, and it was actually on the web, and it was exactly a year from today, last year. We were asked to present information on our product, but since cord blood is cord blood, and all the banks use the same cord blood, essentially, this was a presentation of the safety and efficacy of cord blood in transplantation. So we presented our data, but we also presented data from other transplant centers, and also asked help from other transplant centers to discuss the indications. As I mentioned, it is the regulations that say that the indications for a licensed product have to be specific diseases, so diagnosis. Our position had been all along that cord blood is not a drug, it's a graft. Therefore, the indication is hematopoietic reconstitution. That is a much broader indication than what the regulations would look for, perhaps. But the FDA Advisory Committee was supportive of this, and at the end, in November, we were very happy to see that the FDA approved the broad indication, sort of a shift in paradigm of how we look at the indications for cord blood, and it was approved for hematopoietic reconstitution. That means for any patient that for any reason needs hematopoietic reconstitution. So I hope I convinced you this was not an easy process. It was lengthy, it was challenging. It needed education from both sides. We were not very familiar with the regulations. The regulatory people were not very familiar with cord blood banking and the clinical situation. So we both had to meet the challenge and come together and find the common language. But the goal of all this is to assure that there is consistently high quality of the cord blood units for all cord blood units, so that effectively we improve safety and efficacy of the unrelated cord blood transplants. Well, that was the license, but a very large part of the inventory is not licensed. What happens is the FDA gives a license on a particular day, and only prospective inventory is licensed, not the one that is already in the freezers. Well, there have been thousands of units in the freezers. So, following the regulatory guidelines, we amended the old IND we had, which was only banking, to a phase 2 clinical trial as required by the FDA, and we have opened and is recruiting as a multi-center safety study of the unlicensed cord blood units. The primary aim was the incidence of infection of serious adverse reactions or all infusion-related reactions. The FDA wanted a safety study, so we're there to collect information in all reactions that happen following the infusion of these units. There were data available and we presented them to the FDA, but this is the first time that we're doing a prospective study of the infusion reactions. The indications for the IND product were again the same, hematopoietic reconstitution. So the broad definition of the product and the indications holds both for the licensed product and the IND. And as this IND protocol is going through the various transplant centers and the IRBs, because it has to be approved by every center and become a center program, we get the question very, very frequently, so now that we have license, why do we need investigational products? Why do we have to go through consents? Well, the actual FDA gives the answer for that. This is from frequently asked questions. It's a section that appears on their site. It's still there, and it appeared the first time last March, to say that they realize that the vast majority of the cord blood inventory that is available now in domestic cord blood banks as well as international inventory, they're all investigational products. And the number of the licensed units is very small right now. It will grow as time goes by, but the ability to find grafts should not be limited only to the licensed units. We have the other grafts, and the FDA supports the use of other grafts because this could be, in many situations, the best matches for the patient. But wants these grafts to be distributed under the IND, so under better control. So what are the differences? This is a busy slide, but I thought you might wonder how different are these units? Well, not that different in terms of characteristics. So the license happens on a particular date, and then products that meet the product specifications become licensed after that day. There's some differences shown in red in the product characteristics in terms of potency. There is difference in donor eligibility. The donors have to be eligible for the FDA licensed product. There is difference in the way we can change manufacturing methods and testing assays. They have to be validated. And there is a major difference in the use of this product as a prescription drug, no product consent, versus a product consent in the investigational drug. What is not included here is the expiration date. The FDA mandates that licensed products have an expiration date, but the way we're evaluating the inventory now, we're doing analysis for both IND and license. What is different, however, and I hope that AABB in fact will not be upset with me, is the way that the FDA licensed the bank. They license the product, but they also license the bank. They have to go through a very strict FDA inspection, both of the bank and all the other facilities. The regulations and the quality standards and the quality unit of the bank have to meet all the requirements, and there is mandatory quality release of the product. In addition, any changes in the manufacturing procedures have to be validated. The impact on the whole product has to be evaluated, and they have to be approved by the FDA before implemented. So it's a more strict control of the processes in the bank. And then there is reporting in special ways. So this is the cord blood inventory of the National Cord Blood Program. There are about 58,000 units right now. The vast majority of the inventory is unlicensed investigational units, and the growing numbers of Hemacord, which is the licensed product, are showing. So how can we distribute cord blood for clinical use? Cord blood can be used and is used in current practice for hematopoietic reconstitution. This is the indication. And this product can be either the licensed product or the IND product under the specific protocol with a treatment, with product consent. But perhaps we would like to use cord blood for cell therapies, for the so-called engineered grafts that we hear a lot, either that be ex vivo expansion of the cells or T-cell therapies, other cell therapies that aim against anti-tumor or antiviral treatments. And finally, there is a lot of investigational new treatments that are coming up for cord blood. So how can we bring those to the clinic? Well, again, we can use the IND products from the bank and sort of cross-link, or the word is cross-reference, one IND of the bank to the IND of the particular study, for example, for an investigational study. Alternatively, if we had enough, we could use the licensed products because, as I said, the FDA allows off-label use. So even if this is not for hematopoietic reconstitution, it could be used for the upcoming studies. I'm going to finish with the people that actually did all the work for the FDA and the BLA, as well as the IND. This is the group of the National Cord Blood Program. I am the medical director. Our director of operations, Mike Zanowski, is here, and everybody else we wanted to thank for all the work. In addition, we got a lot of support from the New York Blood Center QA department to go through all these regulatory processes. Thank you.